Hello, my name is Dr. Guzman. My presentation is the use of COMSOL for bioengineering applications. Here at Hofstra University, I'm teaching a course on bioengineering modeling and simulation, which is part of the requirements for Bachelor's of Science in Bioengineering. Bioengineering and biomedical engineering is an engineering discipline for the study of biological entities and materials, with fields such as medical devices and implantable biomaterials. Studying biology and biomaterials is hard because of odd-shaped structures and complex geometries, which are possibly governed by thousands of parameters and equations. Cell culture, materials testing, and animal study is very expensive and time-consuming. Modeling and simulation can address those challenges with the use of COMSOL MULTIPHYSICS computer program for numerically solving partial differential equations using finite element method to determine dependent variables such as displacement, concentration, temperature, velocity, pressure, and voltage. The general steps using COMSOL are listed here, which we will be going over in the next few slides. The outcome of the simulation has to be checked for its accuracy. The first step is to identify the domain or system spatial dimension of operation, noting that 3D is the most computationally demanding. Next is to select the module or top organization level package that contains the bottom level interface. In the example shown here, the solid mechanics interface is selected within the structural mechanics module. Geometries of domains are then modeled using built-in primitives, such as a torus with specified properties and position, and then viewed in the graphics window. Geometries can be imported using other CAD packages. Materials are selected and assigned to the domains. Material properties required for the selected interface are checked. In the solid mechanics interface, density, elastic modulus, and Poisson's ratio are needed. Different physical properties nodes can be added depending on the selected interface. In 3D spatial dimension, domain level nodes in 3D volumes, boundary nodes in 2D surfaces, 1D edges, 0D points are assigned and configured for the model. In meshing, the geometry is simplified and broken down into adjacent finite elements. Each element contains nodes with independent variables xyz position and t time, which are used to calculate dependent variables such as u-v-w displacement. The study can now be executed in stationary or independent of time, or in a time-dependent manner, to compute for the dependent variable solution. If the solver converges by minimizing the error, then results will be obtained. Aside from the primary dependent variable, such as the displacement vector in the solid mechanics interface, other derived properties such as stress, strains can now be displayed in tables and graphs to aid in the interpretation of the outcome of the model. And finally, the simulation needs to be verified, validated, and even iterated for further improvement. Let us focus on the structural mechanics module, particularly the three interfaces. Truss, used for slender elements that sustain only axial forces. Beam, also for slender elements but can bend. And finally, solid mechanics, used for general structural analysis, which allows for the calculation of the stress tensor. Here's an example of a bone osteoblast cell attached on a mineral substrate that is subjected to 200 kilopascal of shear stress. Will the cell survive? What will its deformation look like if the structure is governed by only intermediate filament cytoskeleton? Using the solid mechanics interface, factor of safety is calculated as the ratio of yield and von Mises stress, A value of less than 1 indicate plastic or irreversible deformation, as shown in the red regions in the cell. Substituting the yield to ultimate tensile stress shows that the cell has no regions with less than 1. Hence, the cell will not fail and will survive the applied shear stress. The truss interface is employed to model the skeleton of the cell. This particular design demonstrates that the shear will stretch the cell to less than 100% strain. Doubling the cross-sectional area, however, will lead to reduction of stretching to 65%. Another application of the solid mechanics interface is in materials testing, particularly in the simulation of biomaterial gel compression. This is a more accurate model as displayed by the even bulging of its sides during application of compressive stress, which increases as strain increases. In the gel compression simulation, the gel and flat plates were created as cylinders in geometry with a height of the gel as 3 millimeters and then assigned with respective materials, silicone or PDMS for the gel, and steel for the flat plates. A linear elastic model with a second order partial differential equation, where U is the displacement vector that is solved, is applied to all the three domains. A 2D or surface boundary fixed constraint, where the displacement vector is 0, is applied to the top base plate, since we know that this region will not move during the simulation. Next, we applied another surface boundary, this time a prescribed velocity of 0.1 millimeters per second in the down or negative z direction to the bottom of the indenter plate, since this region pushes the gel downwards at a fixed velocity. The geometry was meshed with normal finite element size that is physics controlled. The study selected was time dependent and evaluated up to 10 seconds with a step size of 1 second. The gel is expected to compress for 1 millimeter or a strain of 33%. After converging into solutions, a surface plot of von Mises stress in a 3D plot group was displayed using the expression solid.Mises, then the plot properties modified, including the color scheme. A deformation plot was overlaid to see the gel compression over time. And finally, animation was exported as a GIF file. To verify the model, the elastic modulus was acquired at a probe point on top of the gel center. The pressure property was obtained at the probe point at different time points and displayed as a table as shown here. The stress-strain relationship is shown to be linear, and the slope was computed to be 750 kilopascals, which matches exactly the elastic modulus of the PDMS material that's used for the gel. Other simulation outcomes are shown here. By varying the applied force vectors, gels will deform differently. The diffusion behavior can be modeled using the transport of diluted species interface found in the chemical species transport module. As a simple demonstration, a concentrated substance found in the red circle diffuses over time in the box with the flux vector represented as streamlines. A concentration gradient across the diagonal of this box can be monitored over time and, as expected, gets more evenly as time progresses until equilibrium is reached. Addition of velocity or u vector to diffusion is called convection. In the top simulations, increasing upward velocity leads to substance being more concentrated at the top of the box. The added fluid flow can also be subjected to rotation, which affects concentration differently. The interface can be used to determine if oxygen coming from the medium reaches the alginate encapsulated cell at the sphere center, and also obtain the maximum alginate diameter to ensure cell survival. In the cross-section of the alginate in gel, the dark blue represents zero oxygen concentration where the cell is located. Hence, the cell won't survive. Using parametric sweep, where the diameter of the alginate is decreased, it was found that 350 microns is the maximum that can support the concentration of 80 micromolar per minute of oxygen required by living cells. In a parametric sweep study for optimization, after study parameters are inputted, the parameter of interest, diameter for example, is varied starting from 1 millimeter to the long axis of the cell at 40 microns with a step size of 50 microns. The heat transfer module has several interfaces, including heat transfer in solids and fluids that can model, for example, the heating of a copper pan filled with water with a heat sink handle. In bioengineering, the bioheat transfer interface can be employed to determine the temperature distribution of blood vessels heated with gold particles, for example, in killing cancer cells. The fluid flow module is also called computational fluid dynamics, solving for the velocity vector and pressure. Here it is shown to model a wind tunnel with a cone obstruction to investigate the air streamlines on a circular cross-section. Visualization of vortex shedding or unsteady separation of flow around a circular body was modeled using turbulent flow kappa epsilon interface. Blood flow to the largest blood vessel, the aorta, can be compared without or with a plaque obstruction. Velocity magnitude can be seen in red for high flow and blue for low flow up to 0. There are more regions of low blood flow in the vessel with obstruction, indicating that there are less nutrient and waste exchanges, which is obviously bad for the cells. The electric currents interface of the AC-DC module enables us to model a steady state Laplace equation, which is the second partial of voltage with respect to position, is equal to 0. A ground of 0 volt was applied to the left and bottom boundaries, and a 110 volt to the circular electric source, and the voltage distribution computed across the domain. In pacemaker design, using an active fixation screw of one of the leads hooked to the heart at an angle with a portion exposed to the blood of the ventricle, the electric current density vector field can be visualized to ensure proper flow of electrons for stimulation of heart contraction. In summary, we have utilized COMSOL multiphysics for modeling and simulation of select bioengineering applications that are used for teaching as well as research, which will ultimately save time, effort, and money. The only downside are yearly subscription and extensive knowledge of partial differential equations and vector calculus, as well as determining the correct biological parameters and simulation conditions. And with that, I would like to thank your participation, and I would be happy to answer any of your questions. Thank you.